So this Jeffrey Jackson is the investigative reporter, at least one of them, for the Highwire. So he wrote a Substack about this, calling it, as you'll see, a bombshell paper. So let's take a 2-minute listen here.
We've talked a lot here about the lab origin, about that conversation. Was it man-made? Did it come from nature? There's a new gigantic bombshell study. I can't overstate the importance of this. And we're going to get to it in just a second because we have to really understand the importance of this. So remember, there's been a back and forth conversation. We had the internal emails from Fauci, from Cowan, from Jeremy Farrar, from Christian Anderson, the virologist, all saying at the beginning of this COVID pandemic, essentially, that look, there's big questions here. This thing looks like there were some lab fingerprints on it, if you will. But we're just going to go out and kind of kill that conversation. But then the big organizations, they all had their own feelers out there. And so you started to see in the early 2023 here, this is the headline: lab leak, most likely origin of COVID-19 pandemic. Energy Department now says, and that's the agency revising its assessment based on new intelligence. And then just a month later, that was in February, in March, just a month later, you have the FBI. They had their own team out there. FBI Director Wray acknowledges Bureau assessment that COVID-19 likely resulted from lab accident. And now the CIA, so this is the New York Post, the CIA had their own people looking into it. They're jumping over. Well, according to this whistleblower, CIA tried to pay off analysts to bury findings that COVID lab leak was likely. Whistleblower. So there's two committees here. There's the Intelligence Committee and the Select Subcommittee on the pandemic, the coronavirus pandemic. And they have both sent a letter to the head of the FBI, Director Burns. And this is a fascinating point here. This is still a developing story. The letter says this: according to the whistleblower, this is a CIA whistleblower, the agency assigned 7 officers to a COVID discovery team. The team consisted of multidisciplinary and experienced officers with significant scientific expertise. According to the whistleblower, at the end of its review, 6 of the 7 members of the team believed the intelligence and science were sufficient to make a low confidence assessment that COVID-19 originated from a laboratory in Wuhan, China. The 7th member of the team, who also happened to be the most senior, was the lone officer to believe COVID-19 originated through zoonosis. The whistleblower further contends that to come to the eventual public determination of uncertainty, the other 6 members were given a significant monetary incentive to change their position.
Wow.
So the CIA is doing what the CIA does, really just manufacture consent here on a small level within that team.
Meanwhile, our YouTube channel and Facebook channel were being taken down, and others like us for daring to report that there were scientists saying that this looked like it was more likely lab origin than natural origin. None of the fact checkers that attacked us or those that took down our pages have ever apologized, said, our bad. As it turns out, we were lied to and we listened to the wrong people. You don't hear anyone on MSNBC or CNN or any of these news agencies apologizing to the people. And here we have all of the top people that were behind the lie, driving the lie. Now whistleblowers are exposing everything.
And it's very important. So remember, we had the original strain, the alpha strain, then the Delta, we were told was extremely deadly, and we had to get vaccinated for this. Kids stay out of school. But then the Omicron strain came. The Omicron is this dominant lineage. And it was first identified in South Africa in late November 2021. And what was interesting about the Omicron strain, well, we'll go to Dr. Valentin Brutal on Twitter. He's a German virologist. And he says this about the Omicron strain: the SARS-2 evolved in complete isolation. That's what he's talking about. While all other variants of concern originated from circulating SARS-2 variants, Omicron originated from a most recent common ancestor that was not circulating anymore for more than a year. And we have a picture here he added to this. This is called a phylogenetic tree. And you can see here at the bottom, you have all of the mutations, all those little dots. They're building upon, mutating. And then this red rocket line comes out of nowhere. That's Omicron. It's not playing with any of those other people. It comes out from early 2020 strain and just shoots over all of those and starts its own tree and dominates the rest of the viral strain. So there was a lot of people around the world going, wait a minute, where did this come from? How is it so potent on the population? It became the dominant. How did it just replace everything else that's been doing its time in nature? Well,
And
some
where did
Japanese
it come from? It was not related to any of the most recent, evolutions. It went all the way back as though sort of it's almost like the Neanderthal man suddenly pops into, is walking down the mall and you're like, wait, well, hold on a second. Where did you just come from? Right? I mean, that's
Right.
what happened with this virus. Right.
Right. And,
Yeah.
there's a lot of theories about that. Well, maybe an immunocompromised person just had it and they were isolated for a really long time, but then they just popped out of nowhere in 2021. But the problem was it was immune to a lot of the antibodies that were out there, which is totally against that theory. So some Japanese researchers started to look at some databases of a lot of the variants. And this is the study. To me, this is like Fauci's original internal email where we finally saw, oh my gosh, there's actually something here. This is a genetic version of that. So unnatural evolutionary processes of SARS-CoV-2 variants and possibility of deliberate natural selection. So they looked at these variants from around the world, a lot in the United States, but some from India, Bangladesh.
Okay, I think that's enough of that. So you hear all the intrigue and the other side being wronged, and all the CIA and the FBI and whoever's get involved. This is just sort of classic things that the people who are criticizing the dominant narrative, and they are being persecuted for their explanation, all the usual stories. But the important thing I want to focus on, as this Jackson says, is this is the bombshell study. This is the study that proves that Omicron is a lab-created virus and will shake the world. So we're going to take a look at that study. So here is the study. Now, before I get into this study, and we're going to take a look at this and see if there's actually any science in this study, let me just get rid of this for a minute. I want to try to compare this to make it easier to understand, which I've done many times before, with how these variants come about. So I've actually written this even as long as 4 years ago, but I think it's good to make an analogy here. I like doing analogies. I think helps people understand. Maybe I'm wrong, but I'm going to try it anyways. And so I proposed that we have a castle that's made of Legos. So this time I actually got a castle. Now, this castle, we're told, is an exact replica of a 12th century English castle. But unfortunately, there's no photographs or even eyewitness accounts of the existence of this castle, or even drawings or any visual evidence of what the castle might have looked like. But anyways, we want to make a reproduction as best we can. So this is sort of the best guess as to what the castle looked like. So the way this comes about is a bunch of researchers were given 56 million different Lego pieces, and they were asked to make this castle. Again, an exact replica of a 12th century English castle for which there's no pictures, drawings, photographs, or eyewitness accounts, or any other accounts of what this castle might look like. Now, you would think that they would ask the obvious question of, well, how would I know what to make? But this is essentially the same with virology. There is no proof that these viruses exist. If you go back to the original coronavirus, which we'll get into that in a minute, and the Baileys again have done a whole piece on the finding of the original coronavirus. So each one refers back to a previous model for the virus. And at no point was that original coronavirus or a coronavirus actually isolated, purified, characterized from nature, the same as this castle. So there's some things to note about this. So notice we have three interesting segments of this castle. For one, we have the sort of blue clock in the front. You can see that. And then the green like thing looks like a little insect or something to my left, maybe your right. And then we have the spire on the top. So we could say these are the three characteristic segments of the castle. And every castle that's in this lineage should have those three segments. And this is similar to what they do in virology. So they don't want to find the whole virus again. They just want to find three characteristic segments of the virus. So they pick these three, and then they do PCR process to find those three segments in all subsequent samples. Now, the question that you would have to ask is, what are the conditions that would tell you that you have the proof that these three segments belong to this castle and only this castle? And here we have a little bit different from my Lego example to what happens in virology, because here we can clearly see that these three segments belong to this castle. The isolated particle called virus, and so never extracted the genome directly from that virus or the RNA directly from that virus. They actually don't know that the segments came from that RNA so-called genome. So there's actually a big difference. So the first condition is you have to prove that there is a castle. Here they have a Lego castle, but you don't know that those three segments belong to the original English castle because you don't have any photograph of the original English castle. So you're using a model of the castle, and then you see the segments, and you're saying that that proves somehow that that model, those segments belong to the original castle, even though you have no original castle to look at. So that's the same with the virus. You have no original virus to look at, so you have a model of the genome, and then you claim that those three segments belong to that genome because they belong to the model that you created, which did not come from the real castle or real virus. So that's the first condition. You have to prove that the segments belong to the original particle, in this case, the original castle, which you can't because you don't have it. And you would have to prove that the segments belong to the original virus, which you can't because you don't have the original virus to evaluate. The second thing is you have to prove that no other castle has these same segments. And so that would be a big job with Lego. You'd have to look through all the other possible castles to make sure that none of them have these three, because if they did, then obviously it wouldn't be enough to make a positive identification of the castle, because if some other castle has these same segments, it could be the other castle. And it's the same with the PCR segments of the viral genome, so-called, because we know that humans have those same segments and other microbes. So there's no way to positively identify that the segments only could have come from that virus, because the same segments are found in other organisms as well. So that makes you have to go back to the original source of the coronavirus, or in this case, SARS-CoV-2, which would be like going back to the original source of the castle, of which you have no picture, no information about. So everything downstream obviously falls apart. So the claim with the variants and with the segments is that they belong to they all emerged from the original genome found for the original or index coronavirus. And this was obviously the coronavirus that was found by the Fan Wu group in Wuhan, based on, we've been over this many times, based on one person. And so they essentially took the secretions of this one person and made 56 million parts of this, and then started to put those little pieces together and assemble them into the viral genome of an organism that they had never seen. And they call this process the unbiased de novo assembly of the genome. This is also known as metagenomics. So they're claiming that there's no bias, that there's no blueprint of which they're working on. And this is the important part of this whole thing. They're claiming that these pieces only fit together in one particular way. And so that way that they fit together represents the genome of the new virus that they found. And this is a claim to be an unbiased process because they're not using any template to tell us what the genome is. They're just seeing where the genome, how these segments, these small pieces, fit together to make one unique genome. Those statements are somewhere between a fraud and a lie, for the very reason that they don't come up with one unique way that these segments piece together. Again, if you remember the Fan Wu paper, there's two crucial points of this. One, they use two different software programs to assemble these pieces into genomes. One program got over 1 million possibilities, and the other got over 400,000 possibilities. So in other words, they took these 56 Lego pieces, and they don't just fit together in this way to make a castle. There's 1 million possible castles that you could derive from those 56 million pieces. Now, everybody needs to be very clear on what I just said. There is over 1 million possible castles that were supposedly derived from those same 56 million segments. So what you need to answer for yourself, if you think about this, is on what basis would you say this is the right castle as opposed to, say, this one? So the 56 million pieces also made this one. And the 56 million pieces also made this one. If you have no blueprint for what the original, authentic, real castle looked like, how would you possibly know which is the correct one? So obviously that's the same question for Fan Wu. How would they know that the 29,000 approximate base pair sequence was the correct one? And there's only two answers to that. One is they made a wild guess, and two, they said it was the longest one and the most like previous coronavirus sequences. Therefore, it's a new coronavirus. By the way, the old coronavirus sequences were made in exactly the same way, so it all goes back to the original. Now, that means it was clearly not unbiased, because they chose the one in a biased sort of way that was the closest, but not that close, match to the previous coronavirus. So it was biased from the start. But there's another central problem with that original Fan Wu genome, which is that when two other assembly professionals, mathematicians, took those 56 million pieces, so now imagine you have, this is your original genome made from somebody assembling 56 million Lego pieces, and they came out with this. And they said, this must be the castle. When we had two other people who are also expert Lego assemblers take those same 56 million pieces, the biggest news, if you want to say the bombshell here, is they couldn't come up with this castle. Let me say that again. They took the original data, the original 56 million segments. They claim they assembled that in their computer, and they got this castle, one of a million, by the way, not unique. And they chose this one because it was the best one, they say. But the reality is there was no reason for choosing this one. And even probably more importantly, you don't actually get this one. So they actually had to put approximately 10% of the sequences manually after the assembly into that original assembly to make it look like a previous coronavirus. In other words, they cheated. And not only did they cheated with a specific purpose in mind, because in these manually inserted extra segments, which are something like 10% or so of the genome. So in other words, 10% of this castle, they actually added pieces which would tell people that it was not a natural evolution from a previous coronavirus, but could only have been created abnormally, artificially in a lab, because they put specific pieces that would actually cue people into that from the start. So they actually engineered from the beginning the lab leak story and embedded that into the artificial, fraudulently made creation that they made in the first genome, which would become the index genome, which would become the template for all further genomes. And they engineered the lab leak theory right into that original paper, that original unbiased de novo synthesis. So this is exactly the opposite of unbiased, and it was actually done with the intent to get people to think that there must be a lab leak, because instead of having, say, this part, they put a whole different part, which they came from a whole different, like, viral line or a whole different castle, just so that people would say, hey, that can't possibly be a normal evolution of this virus. It must be created. And it was created. It was created by the Fan Wu team to fool the people forevermore. And that's where we are 4 years later. Now, how do you get a variant of this? So you tell people, okay, this is now the index genome. This is the blueprint. So forevermore, you take whatever sample you want, some sample from somebody's lung, some sample from somebody who's sick, some sample from a cell culture, whatever sample you want. You break it down into millions of pieces. You tell the computer, make a genome that looks like this. And the computer does that, except it always makes certain mistakes. And so just like the definition of science is that the study needs to be repeatable, they can never actually repeat the genome assembly and get the exact same genome. So it always is a little bit different, just like if you gave people this template and 56 million pieces of Lego, and they weren't very good at it. They would make something that looks like this, but it has a few mistakes, and those become the quote mutations or the genetic variability. And when they're offshoots of the original, engineered segment that wasn't quite right, then you would have some of them that are close, and that's the lower phylogenetic tree that Jackson talked about. So they all clump in one way. And then if you put a whole bunch of mistakes, then you get a whole different phylogenetic tree, which is allegedly the Omicron variant. In other words, the entire reason why you get variants is this is a very anti-scientific process of taking 56 million pieces, giving the computer a blueprint and saying, make the exact copy, because it never does it the same way. The other factor that gets into this, which is not like what happens with Lego, because with Legos, you have 56 million defined pieces if you were going to do this. With making these assembled genomes, you're always using this PCR process. And so it actually makes segments of RNA which actually weren't even there in the original sample. And then when you put in so-called primers and tell it which segments to make, it makes segments along the blueprint of the primers, and then it makes offshoots of that, because PCR always ends up making new segments that weren't there originally, and that becomes another source of the variance. And the third reason you get all these variants is, and this is a little bit hard to explain, but go back to the three segments. In order to say this is this castle, you have to find all three of these segments. But then you say, oh, well, there's a variation of the clock, so it's now a different clock. So that won't show up on the PCR test. So now only two of the three are matches. So, and because we say that's still the same castle, but it now has a variant, from now on, in order to claim it's a variant, you only need two of the three matches instead of all three. And that's exactly what happened with the Omicron variant. They said that one of the segments was so different in its mutations that now the definition of a positive result is the other two match, the third doesn't, obviously because it's different now, and so it won't match. And so now, instead of having to have all three segments as a bogus positive match, you only have to have two segments that are a positive match, which dramatically increases the number of cases. Very convenient for the perpetrators of these theories about viruses and lab leaks, not very good if you're trying to be rational and logical and scientific. So that's another way that they actually play games with how they describe these variants. So to summarize this, it all goes back to, was there an original castle found, or was there original castle in existence, or was there an original virus in existence? The answer is, with coronaviruses, there's never been found. If you go back to the original one in 1968, they never isolated, purified any particle and found any genome from that particle or showed what the particle did. Again, the Baileys have done a very clear and specific piece on this. Everything after that is a template based on that original fraudulent model. And you can see that the unbiased assembly for the SARS-CoV-2 virus was anything but unbiased, and it was based on fitting this into a blueprint of a previous coronavirus, and that didn't even work, so they had to manually do that. And when they manually did it, they added pieces which would be which would tip people off that this was lab created so that we would have two sides of the story, and those would be the only two sides there are. And that's how this whole thing was done. Now, let's go back and see if this squares with this blockbuster paper that they talk about on this Highwire show. So here's the paper. These, of course, when you do this, you always throw out big names. So these are, world-renowned virologists from Japan, and we got the CDC and the CIA and the FBI and all the big shots are involved in this. So what did they do? So the first thing I want to point out, so how do they know there is a genome for the coronavirus? What did they use for that? So here we have reading from the introduction. COVID-19, the coronavirus disease 2019, caused by severe acute respiratory syndrome, coronavirus 2, no reference, or here, there is a reference, was first reported in December 2019 in Wuhan, China, and that is the Fan Wu reference. So it wasn't for no reason that I went into how Fan Wu came up with this SARS-CoV-2 genome and report. This is exactly what they're relying on as their original coronavirus genome. That is the Fan Wu paper. That is the source of all of this rest of this information. And as we can see, they actually didn't find the genome. They can't even assemble that genome from that data, at least nobody else seems to. And they had to put in this 10 or so percent. They had to put it in manually to make it fit. So it was neither unbiased nor de novo. It was basically a scam from the beginning. So let's go on now. So interestingly, like a lot of papers these days, there is no methods section, at least not until the sort of supplement part. So there is a method section, but they don't put it into the body of the paper, which is way different than they used to. But I want to just point out something. So now we have the results, so without the methods. So I want to just read how they did this. So at first, we presumed the existence of the isolate of SARS-CoV-2, whose amino acid sequence of S protein contains the Omicron BA.1 type mutation subsets, but one mutation position was not mutated and constituted with the original Wuhan type amino acid sequence. So the question that we need to ask about this paper is actually, did they do any biology? Did they do any virology? Did they isolate and purify anything? Did they, as one would expect, find these particles called coronaviruses making certain people sick, isolate them, purify them, find the genome of these, find that it was different than the original one in a very specific way that would lead you to think it's its own evolutionary tree and a actual specific variant? And the answer is, of course, no. They didn't isolate anything. They didn't work with any biological sample ever. And as you see, they only presumed the existence of the isolate of SARS-CoV-2 in the first place. Once you presume the existence of something, so if you presume the existence of this Lego castle and it looks like this, and then you make it and you say, see, it looks like this, obviously that is circular reasoning nonsense. You haven't proved anything. You've just made a claim that you assume, if this is real, that this would be a reproduction of it. But the entire issue is whether that SARS-CoV-2 is actually isolated and studied, and they only presumed the existence. And so now we go into the actual methods section, which again is in the appendix, not in, or the supplemental part, not in the body of the paper. And let's read some of this. So what they did. Okay. SARS-CoV-2 RNA genome and genes and proteins according to an annotation of SARS-CoV-2 Wuhan Hu H1. That's the first isolate. Again, they're referring everything back to the Fan Wu paper. That's why in order to understand this, you have to understand that Fan Wu never came up with the genome of anything, certainly not a coronavirus, and they inserted pieces which would actually tell people that it was created in a lab, or not tell them, fool them into thinking, because they put in segments that were inappropriately matched, so to speak, for a natural evolution. So in other words, they embedded this story, somebody from the very beginning. These reference segments were used as a reference strain for the definition of mutations and provided base of the numbering of the nucleotide and amino acid numbers of each protein. In other words, they used that same genome for all the subsequent information for definition of mutations, etc. Genome data of SARS-CoV-2 isolates, which showed in this article, were obtained by copying from the NCBI nucleotide database. So in other words, they didn't find anything on their own. They didn't isolate anything. They didn't find any particle with a different genome. All they did was downloaded stuff from a database. So this is not virology. It has nothing to do with biology, or this is basically computer technology, so-called science. This is just computer database stuff. And if you don't believe me, amino acid sequences of spike protein of SARS-CoV-2 variant, all these different ones, were obtained from the internet site Stanford Coronavirus Antiviral and Resistance Database and used as a query sequence for the NCBI protein blast search. So in other words, all this paper, this blockbuster paper, is looking at previously downloaded sequences that were all based on the original fraudulent Wuhan sequence. Never anybody along the way showing that any of these sequences actually came from something called a virus. So it goes on. The whole genome sequence of each variant isolates bearing the query spike sequence were derived from the blast search resultant, which identified with query amino acid sequences with 100%, and then the nucleotide sequences of detected SARS-CoV-2 genome were as follows. And these are all GenBank ascension numbers. And so then the rest of the methods is listing these various downloaded sequences from the GenBank. There's no mention of here of any biological anything, no culture, no isolation, no purification, no characterization, no finding of any particle ever. It's simply just computer technology, all based on the original fraudulent genome as I described. So, and here you can see the rest of it. It's just a bunch of sequences. And so that's the blockbuster paper. So to summarize, since one way to understand this is to compare it to a Lego castle. And as we say, just like the coronavirus, at no point in the lineage was the castle actually positively ID'd. The same with the coronavirus. At no point in the history of coronavirus research was an actual particle obtained and a piece of it found to be the genome. That never happened. With SARS-CoV-2, the so-called de novo unbiased sequence was anything but unbiased or de novo. It was made in order to instill or put this story of the lab leak into the world discussion, so that we would have two sides of the two sides of this conversation. Either it's a natural or a lab leak, when the reality is it's neither. There is no blockbuster here. There is nothing to see here except the usual virology nonsense. So I would then finish with, why is this important? And to me, if the people who are waking up in the so-called truth and freedom community, if they don't get with the depth of the program that's being run here, it is not about a lab-created virus. It is not about reforming government agencies so they don't do this. This is about a fundamental shift in the way we see the world. And we cannot let ourselves be put in this box of, here are the two possible solutions. It's either this or that. That is just still a inhabitant of this matrix, which is the source of our difficulties. We need out of the matrix. We need to see this world in a different way. And I will finish with saying, if either Big Tree or Jackson think that I'm incorrect about any of the facts that I say about this paper or the Fan Wu paper, I would appreciate if they would send me or do a public refutation to describe actually how they know that this virus actually exists, and the genome comes from that particle that has been isolated and purified, and not in exactly the way that I described. And my guess is that will never happen, because it can't possibly happen, because I am convinced that my analysis, maybe not right in every single detail, but is largely correct. And this is exactly how it came about. And we cannot go on and let the people who are otherwise waking up to this fraud not to see the enormity and the depth of this fraud, because it's only when we see that we really liberate ourselves from this awful mess that we've gotten ourselves in around virology. So again, thanks everybody for listening, and hope everybody stays off their computer from 2 to 4, and I will see you next week.